Hi. So, we have studied a lot of pattern recognition receptor, and if you see your previous sessions, we have started this pattern recognition with discovery component. We have learned how the pattern recognition receptor concept was emerged, and thereafter we have learned how this pattern recognition receptor was discovered. The first pattern recognition receptor was a toll-like receptor, and we have extensively studied toll-like receptor. Then we have studied discovery of Rigi-like receptor, and then we have also studied various signaling pathway, the disease associated with RLR pathway, and we have also studied the node-like receptors, and we have again studied the various ligand, the signaling pathway, and the disease associated with node-like receptors. So, today we will take up the last pattern recognition receptor, and this pattern recognition receptor is a DNA sensor. Okay. And this DNA sensor if you see, it's very important in sensing various DNA viruses. Okay. So, let us begin with the discovery of DNA sensor, and if you see this slide, you will understand that the immunostimulatory property of DNA in order to induce antiviral response was kind of discovered or noticed by the scientist about 50 years ago. Okay. And this is basically started with one simple observation. When mammalian cells are when we introduce the DNA in mammalian cell, then that induces some kind of immunostimulatory responses. Okay. So, if you see carefully the pattern recognition receptor, toll-like receptor, so there is a one TLR which is sensing the DNA molecule, and that TLR is TLR9. Okay. And this TLR9 basically TLR9 is also a DNA sensor, and this TLR9 basically senses hypomethylated DNA molecule, particularly CpG region in DNA, which is quite abundant in viruses and microbial cells, irrespective of pathogen or non-pathogen, compared to the mammalian DNA. Okay. So, in general, the mammalian DNA is quite heavily methylated, and that is why they are not sensed by the TLR9. So, this is the original discovery of TLR9. Here you can see that this toll receptor basically recognizes the bacterial DNA. And if you just carefully see, Professor Shizuo Akira is a person who has as I told you in when I discussed the discovery of this pattern recognition receptor. So, Professor Shizuo Akira, he basically characterized most of these TLR sensors, various adapter and signaling molecules. So, this is a one of a very good example. He basically characterized he and his group characterized this TLR9. So, people observed that this TBK1, if you remember the TBK1 in TRIF dependent signaling. Okay. So, TBK1 is associated with IKKI or IKK epsilon. Okay, this kinase basically phosphorylate IRF3 or IRFs, and then that induces the production of type 1 interferon. Okay. So, people observed that the induction of type 1 interferon is basically dependent on TBK1. Okay. And this suggests that okay, the signaling axis, the TBK1 IRF3 axis is same as TRIF dependent signaling or RLR signaling. Okay. So, this signaling is or this signaling axis, this TBK1 IKKI IRF3 signaling axis is also needed for the DNA dependent induction of type 1 interferon. Okay. So, it is a very simple experiment, you can very easily prove it if you have a TBK1 knockout mice. Okay. You simply introduce this poly-DADT or double stranded DNA. Poly-DADT is a synthetic ligand which commonly used for activation of DNA dependent signaling. Okay. So, if you introduce this poly-DADT inside the cell by chemical mean or by electrochemical mean, then you will observe that there will be induction of type 1 interferon, but this production of type 1 interferon will be completely abolished if you use the TBK1 knockout mice. Okay. So, this observation was quite well taken by the scientific community, and then this studies suggest that there are DNA sensors which is upstream to the TBK1, and that DNA sensors basically bind with DNA molecule and induces TBK1 IRF3 dependent type 1 interferon. Okay. So, this is this simple experiment also creates a question in innate among innate immunobiologists. Okay. And that result to the discovery of DNA sensors. Okay. So, the first DNA sensor which was reported in around if you see carefully, it is for the first DNA sensor was reported in 2007. Okay, and this is the DNA sensor. So, this group, this is again Japanese group, this Taniguchi group discovered the or reported first cytosolic DNA sensor or receptor. Okay, and they gave this name DAI. Okay, DNA dependent activator of IRFs. Okay, so this name was given by this group to this DNA sensor. This is also known as DLM and ZBP1. Okay, and this work was quite well established that okay, there is some DNA sensor, and this DNA sensor can sense and induce TBK1 dependent and IRF3 dependent induction of type 1 interferon. So, this DAI sensor is also inducing NF-kappa B activation in order to induce inflammatory cytokine. Okay, after binding with the DNA. Okay. This was a first discovery, the discovery of DAI. Another concept was emerged that you know that genome, our genome is having 80 rich sequences. Okay, and it was suggested that RNA polymerase 3 is capable of transcribing this 80 rich double stranded DNA. Okay. and this basically creates RNA molecule, and that 50 around 50 base pair RNA molecule is having this triphosphate motif, and this triphosphate motif, if you remember, can activate the rigi. Okay. So, this work basically suggested that DNA is sensed, but it is indirectly sensed by rigi. So, there will be a poly-DADT, and this poly-DADT is first transcribed to the RNA molecule, which will have this triphosphate motif, and this RNA molecule with this triphosphate motif will be sensed by rigi, and then there will be a TBK1 IRF3 dependent production of type 1 interferon. Okay. This was basically supported by two research paper. If you can see the middle panel as well as the bottom panel, this which suggested that RNA polymerase 3 plays a very important role in DNA sensing, indirectly DNA sensing. Okay. But we have there is a report that showed that this DAI is not playing important role in vivo condition. Okay. In vivo condition, they do not sense the DNA vaccine, okay, and they are not important in production of type 1 interferon. So, this can be very easily achieved. There is a simple experiment. If you have a DAI knockout mice, and if you stimulate with a DNA molecule, then if it is still inducing the production of type 1 interferon, then that will suggest that so DAI is not very important. Okay. Maybe in some cell type it is maybe playing an important role, but in general in vivo under physiological condition DAI is not very crucial. Okay. So, here you can see that there is a work that tank binding kinase 1 delineate innate and adaptive immune responses to DNA vaccine. So, this work basically suggested the same. So, DAI is a cell type specific, and that polymerase RNA polymerase 3 rigi pathway is only detected AT rich DNA, and this work, all these work clearly suggests that there must be another or additional cytosolic DNA sensors, okay, and that need to be discovered. So, in research, giving a question is also very important, or giving a direction to the research is also very important. So, all this work gave a direction for the discovery of DNA sensors. Okay. So, after that there was a discovery of one molecule which is basically a adapter protein called as a sting, and this sting, when this sting was discovered, simultaneously this molecule was reported by several other groups, and they gave different name like MITA, MPYS, ERIS. So, all these names were given independently. Okay. When a sting was reported, simultaneously the same molecule were reported by other group with different name, and people showed that this sting directly engage or activate TBK1, okay, upon DNA stimulation. So, this suggested that this sting is playing very important role in activation of TBK1 and subsequent activation of IRF3. However, this sting is not directly binding to the DNA molecule, suggesting that this is not a sensor. Maybe this is a one of signaling transducer, okay, signal transducer of this DNA sensing pathway, and people suggested it as a adapter. Okay. So, there are several papers which was published almost together, here you can see, and all these work suggests that sting is playing an important role in DNA sensing. However, it is not directly interacting with DNA molecule, and it is inducing the type 1 interferon depend on TBK1 and IRF3. So that basically is a very important or very key finding after DAI. DAI was not successful in vivo condition. So, sting was a quite correct discovery, and this plays a very important role, and this is playing an important role upstream to the TBK1. Okay. So, since then a lot of work was performed, and then there was a discovery of various DNA sensor, and here you can see that since 2009 at least 10 protein have been proposed as a cytosolic DNA sensors. Here this is a very nice schematic also, and those molecules are AIM2. If you remember AIM2 in NLR. So, AIM2 basically bind with the DNA molecule, and it induces IL-1 family cytokine. Okay. IFI16 is also playing a very important role in induction of type 1 interferon and inflammatory cytokines upon stimulation with DNA. Okay. And other molecule like LRRFIP1, DHX9, DHX36, DDX41, KU70, DNA-PK, MRE11, and C-gas, STING, and RED50. So, all these are several DNA sensor, and here you can see the the discovery of these molecule in chronological order. If you see this graphical representation, the first DNA sensor was which was discovered and well documented is TLR9. After that there is a discovery of DAI. However, it was suggested that in vivo they may not play a very important role. After that AIM2 was discovered, involvement of RNA polymerase 3 was reported, and subsequently various DNA sensor was reported. And all these DNA sensors basically play a very important role in induction of pro-inflammatory cytokine, which is NF-kappa B dependent, and production of type 1 interferon. In next session I will talk about the DNA sensors and what are the ligands and what kind of signaling pathway they activate. Thank you very much.